that we had talked about last time around? Anybody, any questions, comments, anything?
So, while reading the papers, I found that there are multiple types of epistasis, right?
Correct.
So, can you shed some light on how many types there are?
Yes, we will do that today. Yeah. Yes. So epistasis is a broad term which simply means that two mutations are interacting with each other and then having an effect. But those interactions could lead to those interactions could mean different things in terms of functionality and fitness of the organism, and that classification is something that we'll talk about today. Yeah. Anybody else? All right. Okay, let's start then. So that's it. In fact, that's what I want to do before we pick up our discussion on allele frequencies. Excuse me. So this is just a short note on epistasis that I wanted to discuss a few things. And as you've seen from the two papers, that epistasis is actually a very pervasive phenomena. Uh, it's present everywhere, and epistasis is complex because of the following reason: that if you were to give me two different loci, and we were to try and understand what is the nature of epistasis at these two different loci in the chromosome of the organism, we would probably be able to find that out. We will find out what genes these two loci belong to, what are the functions of these genes, are they interacting, are they part of the same pathway, do they catalyze reactions which lie in the same pathway. And once we understand the underlying biochemistry of what these loci were doing, we can find out the nature of epistasis between these two specific loci. That's the part of epistasis that's sort of well understood, and that's where we are in terms of the field in trying to understand how do we actually perceive epistasis. The challenging part about epistasis is that how do I understand epistasis at a global phenomena? What is the degree of epistasis? I mean, is most of the epistasis of the nature where two different loci are interacting with each other, or what is the degree of epistasis? That means, are there, like, on an average, are there several loci which are interacting with each other, or epistasis is mainly pairwise where two different loci are talking to each other? And what are the global patterns of epistasis? uh, if I were to look at cell as a whole and not look at two specific uh, loci, and the interactions between those two loci. So some of these questions about global patterns of evolution are actually global patterns of epistasis in a cell are actually just beginning to be understood. And if anybody is interested, I can share a couple of references from just last year, which tell us a little bit more about these things. I mean, I think in what I have said, I have not been extremely clear because uh, it's a difficult thing to explain without really getting into the details, and I don't really want to do that at this point. But if any of you are more interested in sort of the larger context in which epistasis is being studied now, just write to me and I can share a couple of references. All right. So epistasis is actually a term first coined by Bateson, and Bateson, remember, was from the Mendelian and biometrician debate. Uh, and what he noted was that some mutations stop or stand above other mutations. That means there is a mutation A, which, when it happens, so there is another mutation B, which has a certain effect on the organism. But if mutation A happens, then whatever happens to mutation B, if mutation A is present, then irrespective of the fact whether you have B or not have B, it makes no difference. B doesn't manifest itself if A is present. So in that sense, mutation A is standing above or stopping mutation B. And from this, Bateson concluded that mutations were actually interacting with each other. And he said that such mutations are said to be epistatic, the ones which stand above other mutations, which stop functionality of these other mutations. And the ones which do get stopped, he referred to them as hypostatic. Fisher is the statistician whose work we'll be looking at next week. He added that epistasy is simply the deviation from the additive combination of the two loci in their impact on the individual phenotype. So he's saying epistasis as the following phenomena. If you have mutation A, then you are 1 centimeter, you gain 1 centimeter in height. If you have mutation B, you gain 0.5 centimeters in height. If you have A and B, the null model will be that you gain 1.5 centimeters in height. Any deviation from this gain of 1.5 is epistasy as far as Fisher is concerned. If you're carrying both A and B mutations, and you gain 2 centimeters, then that additional 0.5 that you have gained is the deviation from the additive combination of the two loci, and that is a phenomena that Fisher says is epistasy. And of course, epistasis is studied in many contexts. But one of the really striking phenomena is this paper that was published in 2007, and I'll share it on Moodle. It's actually a fantastic example of how epistasis and evolution can work. So what the question at hand is sort of the evolution of receptor for cortisol in all vertebrates. Okay? Cortisol is a steroid hormone. Cortisol is just a steroid hormone, and all our cells have cortisol receptor. Okay? And cortisol is produced in adrenal glands in our body, but its implications are extremely large, ranging from controlling metabolism to stress response and many other functionalities. So what the paper is about is the evolution of receptor for cortisol in vertebrates, right? And what they find, and they do a large scale analysis of existing cortisol receptor sequences in different vertebrates, and using that they reconstruct a cortisol receptor sequence, and they call it a ancestral protein sequence. And how they get to that is sort of not very important, but for our purposes what we will take it to be is that there is some ancestral sequence, right, of a receptor in a cell which can sense cortisol. Then they basically identify two mutations, right? And those two mutations are, so there are 20 different amino acids, and a standard way to represent a mutation is that in this ancestral protein sequence. So before I do that, let me tell you, this ancestral protein sequence, if we were to actually make this protein sequence and put it in an organism, we will find that it's very promiscuous. It is not very specific to cortisol, and it interacts with all sorts of ligands. So what we would like is this ancestral protein sequence to evolve in a particular way to acquire mutations in the sequence such that it becomes specific and very high in activity as far as sensing cortisol is concerned. That is the goal. And starting from this ancestral sequence, which was very promiscuous, it could sense all sorts of ligands. What they find is that there are three classes of mutations that can actually happen. The first class of mutations, they call group X mutations. These group X mutations, what they do is that they make the receptor protein a somewhat more specific receptor for glucocorticoids, of which cortisol is one particular example, right? So this mutations of group X make it more specific, right? Earlier it could recognize anything, but specificity is important in biology, and via acquisition of these two mutations which belong to group X, we make it more specific for glucocorticoids. So that's one step towards specificity. Of these two mutations, there are two, and a standard way to represent a mutation is that you have an amino acid that was present at this particular location, right? Uh, and because of this mutation, this amino acid was changed into this amino acid. So there are 20 amino acids, and each one is represented by a letter. S, for instance, is serine. You can just look it up as to which amino acid stands for what alphabet, but that's not really very important in the context of this particular discussion. So these two mutations enable it. Interestingly, that when you have one of these mutations, L at the amino acid L at position 111 changed to amino acid Q, it has no effect on the protein function, right? So this mutation by itself doesn't do anything. On the other hand, the other mutation, S at position 106 being changed to P, destroys function by itself. So this ancestral protein sequence could basically sense a lot of ligands, but if you were to this sequence, if you introduce this one mutation, S106P, then it destroys function, it doesn't sense anything. This other mutation does nothing. But when they are both present, you get a receptor which is more specific towards glucocorticoid sensing, right? So that's within group X, you have epistasis happening. That one mutation does something, the other mutation does something, but when you put both of them together, the outcome of the presence of those two mutations is something completely different than the additive effects of those two mutations. Does that make sense? Any questions with that? All right. All right, cool. Then there is another class of mutations which they call group Y mutations, and these are 3 in number. When these 3 mutations occur in a group X mutant sequence, so these two have already happened, so we already have a receptor which has been made more specific to glucocorticoid sensing. When this particular double mutant carrying 2 mutations of group X additionally acquires these 3 mutations in group Y, and I've left out the details here, what this does is that it makes the protein specific for cortisol reception, right? So among glucocorticoids, now we have a protein which is very specific for cortisol, but it destabilizes the protein a lot. So if you have a XY receptor, it's very specific to cortisol, but it's extremely unstable, right? So you're probably not going to get very much out of that too. Third set of mutations is another 2 mutations which they call group Z. And what this does is that now if you have X mutation and Y mutation, and if you have a mutant version of the receptor protein which is carrying the mutations of group X and group Y, if you were to additionally acquire these 2 mutations of group Z, then it would stabilize the X and Y mutations, meaning that now you have a mutant version of the protein which is only specific for cortisol and is also stable. So you sort of arrived at the desired protein sequence. Now, what we are going to do is look at these mutations in the context of just groups. We'll forget about this epistasis that exists within groups. But if we have mutations in X, Y, and Z, is there a particular order in which these mutations need to happen as far as evolution is concerned? One group makes it more specific for only glucocorticoids. Second group makes it very specific for cortisol, but completely destabilizes the protein. And the third group stabilizes the protein. Is there a particular order in which these mutations need to happen? Is the system we described clear?
Yes, sir.
Okay.
So I think there needs to be a sequence of mutations.
Yeah. Yeah, and it's not completely obvious as to what that sequence might be, but here is this paper, and if we look at that, I mean, it's a lot of details, but if you look at this particular figure, right? So what we are looking at is the receptor sequence and its sensitivity, some measure. We're not going to go into the details of what is this measure, but we have three hormones here, and what we are measuring is that for this particular sequence, what was the sensitivity for each of these three hormones. This is the ancestral starting sequence, and it had some degree of sensitivity. Forget the numbers. The point is it's non-zero. We can detect this sensitivity. So for these three hormones, there is some degree of sensitivity associated with the ancestral sequence. So very promiscuous, as we said that the ancestral sequence is low level sensitivity for a large number of ligands, right? So it senses everything. If you acquire mutations X, you basically become more specific for glucocorticoids, of which these three are examples, right? So your sensitivity for each of these three increases. If, however, you only acquire Y, there is nothing, right? Because remember, Y only made it sensitive for cortisol, but only if you had X, right? So just introducing Y by itself, the set of mutations, three mutations in group Y, doesn't do anything. If you introduce both X and Y, now this is interesting because this is very specific for cortisol. However, it basically is it's an extremely unstable protein molecule, and hence you don't sense sensitivity in this particular assay. But they figure out using other assays that this is in fact extremely sensitive to cortisol. In this background sequence, if you were to only introduce Z, whose job, remember, was to stabilize the protein only when X and Y were present, it doesn't really do anything if you introduce Z by itself. If you introduce X and Z, you see a huge increase in activity, in sensitivity of sensing 1, because remember, X only makes it specific for glucocorticoids and not cortisol per se, right? So XZ is very sensitive to aldosterone, but not the other two. Similarly, YZ, if you acquire YZ, you are then specific to 1, and if you have all three, then you become very sensitive to cortisol, right? So if we define evolution of this enzyme, or evolution of the individual or a population that is carrying this enzyme, and we define fitness as sensitivity to cortisol, okay, if that is what we are doing, that is our measure of fitness, then if in this background we introduce mutation X, sensitivity increases, and hence mutation X, again these are not individual mutations, X is 2 mutations, Z is 2 mutations, Y is 3 mutations, but let's not worry about that detail. If the mutation X happens in the ancestral background, then you have increase in sensitivity, which means X was a beneficial mutation. Does that make sense to everybody?
Yes, sir.
Right? All right. However, if we have X take place, if we have mutation X take place in a background of ancestor plus Y, nothing changes. So X actually becomes a deleterious mutation. I'm sorry, a neutral mutation, because it was 0 to begin with, sensitivity, and it was 0 after introduction of X. Look at this then. If you have a version which is carrying ancestral plus Z group of mutations, and to this background you introduce mutations X, sensitivity actually goes down, right? And what that means is that if the same mutation X was introduced in this particular, if the group of mutations X was introduced in this particular background, then the mutation X actually becomes a deleterious mutation. So in this very small prism of protein evolution, what we are seeing is that mutation X can be beneficial, it can be neutral, it can be deleterious. Right? And what this does is that it limits sort of the evolutionary paths that you can take as you are going from this particular sequence to this particular sequence. Remember, Darwinian evolution will only take place through paths. Darwinian evolution will only take place through paths where fitness increases. If you have fitness decreasing in going from one particular sequence to another particular sequence, then in a Darwinian sense, that path is not permissible, right? Because taking that path to go from this fitness to this fitness, if that requires even one step where fitness actually decreases, then natural selection will not permit it, and hence this path is not permissible. And it's the same thing happening here. So what would be a good exercise is to understand that if we are starting from this ancestral sequence, which was able to sense all three hormones, if starting from here we want to end up at this particular location, so green hair, so I'll just give you the logic in which this is presented. Green hair means that this particular sequence is able to sense aldosterone. Purple means that it's able to sense cortisol, and orange or yellow means that it's DOC. It's able to sense DOC. So if you have all three, that means this particular sequence can sense all three, right? So for instance, this is plus Z. So starting from this sequence, you acquired mutations of group Z. And as you go from group Z, sensitivity more or less remains the same. Hence, this is what the authors are calling it, a more or less a neutral change, and hence fitness doesn't really change, and hence this is indicated by a plain arrow. However, in this Z, if you acquire X now, so in this Z background, if you acquire X, then at least for two of these receptors, sensitivity, two of these hormones, sensitivity increases extremely highly. So this is going to be a change, and presumably you need this sensitivity for some physiological function. So this change, this acquisition of X in this particular background, that is represented by us, that is sort of a beneficial change that has happened in the sequence of the enzyme, and hence that move is represented by this green arrow, right? And you can still sense all these sequences, can still sense all three of these hormones. This one can do it, this, of course the extents are different. So that is the thing. And whenever you have this red block here, this is a move that's not permissible, because you going from here to here, you stop sensing all three. So it makes no sense for you to evolve in this particular direction. And hence this is a path that is not permissible as far as Darwinian evolution is concerned. So the idea is to go from here to here, and it's epistasis which basically limits the paths that you can take to go from one sequence to another. And this particular one, I mean, Thornton's group it does a lot of this kind of work, and this particular example is a very striking one of how epistasis can shape protein function. Does that make sense?
Yes, sir.
All right. So your question, Yash, to that effect, imagine if we have a original sequence of a protein, right? And if an individual is carrying this particular sequence, it exhibits a certain trait value. Let us call that 0, right? It doesn't need to be 0, but we can normalize everything. So we can normalize everything with respect to the original sequence and call it a 0. If you are carrying mutation A, then the trait phenotype is PA. If you're carrying mutation B, it's PB. And if you're carrying both A and B, then the phenotype is PAB. And this sort of gives us the different types of epistasis that can happen. If PAB is simply equal to PA plus PB, then it is simply called additive interaction, and there isn't any epistasis, because the effect of carrying both the mutations is simply addition of the effects of the two individual mutations. If PAB is more than PA plus PB, so the combined effect of both the mutations is more than the sum of the effect of the two individual mutations, that is referred to as positive epistasis. If PAB is greater than PA, PAB is greater than PB, so this bar here is greater than both of them individually, but is less than PA plus PB, that is called negative epistasis, and this is the effect that you saw in the 2011 paper. On the other hand, if PAB is less than 0, so for some reason, if you were carrying one mutation, it was beneficial, carrying another mutation, it was beneficial. But if you were carrying both mutations, then the fitness effect was actually detrimental. That is called sign epistasis. So these are the classes of classes in which epistasis can take place in a protein and consequently the organism's fitness. All right, so that's epistasis. Any questions with this?
So, in case of sign epistasis, is it that two beneficial mutations will become deleterious when they're combined, or can a deleterious mutation become beneficial in case of, like, when it combines with another deleterious mutation?
Yeah. So
Or
what?
like,
Yeah, absolutely, it can. So what I have shown here is the manifestation, is the example of epistasis where both these were beneficial, right? Similarly,
okay.
similarly, you could also have if two were deleterious. So
Okay.
all of this also holds for deleterious mutations also. So if you had two mutations which
Okay.
were deleterious, uh, and when you combine them two together, if the net outcome is now increase in fitness, that is sign epistasis too,
Okay. Okay.
right? And in fact, that's seen a lot because this is, I mean, classical genetics. That's an the example that you cite is done a lot, because imagine a protein sequence and you have one mutation that happens at one particular so this is a protein sequence, a mutation happens here, and this, because of this mutation, functionality is lost. So this mutation is now deleterious in nature, right? Now what you will do, what a classical geneticist will do is will sort of mutate this entire sequence, will try to identify what is called a compensatory mutation. Okay, will so let me rephrase that. You have a protein sequence, it is functioning, it's fine. You introduce this one mutation and functionality is lost, Hence, this becomes a deleterious mutation. But now, what people will try and do is try and mutate this again such that functionality is restored. Okay, and there are two ways that this can happen. One is that this mutation can go back to the original sequence, right?
Yeah.
Right, and functionality
Yeah.
is restored. But that's not really an interesting case because you just went back to the ancestral sequence. The other
Yeah.
kind of mutation is that somewhere along the sequence of this protein, you picked up another mutation here. And when, remember, all these mutations are being picked up in a background which is already carrying this particular mutation. So there is no functionality. But if in this background you picked up this additional mutation, then you have functionality restored. So it was almost as if this mutation made the functionality go away. But if along with this mutation, another mutation happened here, functionality is restored. So this one is called a compensatory mutation. Okay.
Okay.
So now
So
these two, so then you can go back and check that what happens in a sequence where only this mutation is present. And invariably, what would be the case is that this is also deleterious. Okay. This was only muted because it was happening in conjunction with this particular mutation. And in terms of protein sequence, think of it like this, that a mutation happened somewhere such that the protein structure got altered in a way such that maybe the catalytic site of the enzyme was changed. Something changed, right?
Oh,
Now,
okay.
in order to restore the site of the site of where the reaction is going to take place, you need another mutation to take place, which would restore that site of catalysis. Right? So that's an example of sign epistasis, where both were deleterious, but when both together, it actually becomes beneficial. Yeah, Anirudh, go ahead.
Yeah. So in the first paper, I think they mentioned that um, that the rate of mutation is very low as compared to that is the time taken for a second mutation, for example, is this is an assumption they've made that is I think is much greater um, than the time it takes for a particular mutation to be fixed or removed.
Yeah.
So I don't. Would it be possible then in a natural scenario that A and B would be found if both A and B
Coexisting.
are deleterious? Yeah,
Yeah.
because
Yeah.
if A comes out, then it wouldn't allow B to come along with A, the mutation
Correct.
along with, because A would be deleterious.
Correct.
That's
Correct.
true.
It's not actually.
That's not.
So yeah. So the first paper is by Daniel Hartel's group, and Daniel Hartel is an extremely famous population geneticist. Uh, Let me pose the following two scenarios. The first one is, and actually, if you look at the schedule of how this course is structured, in I think 3 classes or something, we are going to be discussing what is called a traveling wave model of evolution. Okay, this was a paper that was published in 2008 by a couple of guys at Stanford. Uh, and the issue is this, that you spoke of two time scales. The first time scale is a waiting time for a new mutation to occur, right? And the second time scale is the time it takes for a beneficial mutation to go to fixation, right?
Or
Now,
or, uh,
what
or
is it?
deleterious mutation to be removed from the population,
Correct, correct.
right?
Sure. Sure. Uh, it turns out, and we will do the math again in a couple of weeks. Uh, I think it's the last class before the midsem exams. It turns out that what do you think is a typical size of a bacterial population? You're growing bacteria in a flask. Give me a typical population size.
1 million bacteria.
10 power 9, 10 power 6.
6 is too small.
Okay.
Right? 9? Do you grow? I don't know if you grow bacteria.
Right.
6 is too small. Typically, if you're talking rich media, you're talking of 10 power 9 bacteria per milliliter. Okay? So if you're growing 100 milliliters of LB culture, you're talking 10 power 11 bacteria, but 10 power 9 is good enough, right? Now, the relative lengths of, so let's call the waiting time as tau 1 and the time to either go to fixation or extinction as tau 2. Okay. Now, the relative durations of tau 1 and tau 2 are actually dependent on population size, because the waiting time is a strict function of how many individuals you have. The more individuals you have, the smaller the waiting time for newer mutations to crop up. Does that make sense?
Yeah.
Right? So it's a function of n. N is the population size. And what was sort of discussed in this 2008 paper that we'll talk about is that for bacteria, it is almost impossible for you to design an experiment where tau 2 is what do I want to say?
Is comparable to T1.
Yeah, so in all typical settings, tau 2 is much greater than tau 1. Okay. So the waiting time, because the population size is so large, the waiting time is much shorter as compared to the time for fixation or extinction. What the consequence of that is that if you were to take a snapshot of any bacterial culture at any given time, you are going to see many different alleles at any locus coexisting at the same time. Does that make sense?
Yeah, but in that case, for example, one bacteria should be having both the mutations, right? PA and PB. So isn't that so you're stating that that is more likely because of the size of the population?
No. So not one carrying both. That's not what I meant. I just meant that A and B are going to coexist in the population.
Right, but aren't we talking about when both?
Mm.
So in your previous scenario, you mentioned that, for example, if both A and B were deleterious, then based on what their assumption was, you would never have A and B
Okay.
in a
Yeah.
single population. So by the
You mean a
time,
single individual,
yeah,
not
because
population.
A would be
Yeah.
deleterious and B
Right.
would be deleterious, so it
Correct.
would never come to the scenario of A and B.
Yeah. Yeah.
That's what I was thinking.
Yeah. Sure. No, so again, I think it depends on the precise ecological context in which this is done. Assume that when A happened, maybe that functionality wasn't really essential depending on the ecological niche, right? But if your population is very large, so you have a population where everybody is carrying A because it's not needed, okay? Uh, because that functionality is not needed. However, if your population is, so we can think of it like, we can think of this as resistance to antibiotic. Maybe the antibiotic wasn't there and everybody was carrying mutation A, which was a deleterious mutation, which in a bacterial context means that you are sensitive to the antibiotic, but the antibiotic wasn't there, so you didn't care. But if the population was really large, then there is a chance that one of the individuals was carrying mutation B also, right? So you have one individual in the population which is carrying A and B, but still no antibiotic. And now should an antibiotic come, everybody is going to be wiped out and only that one individual will be left. And that happens because of that epistasis between A and B.
Right. I'm sorry.
Right?
I think I was thinking of mutations happening during the time of stress, for example, during.
Yeah. No, it's not going to work out that way. Again, it also depends on, uh, the context. Uh, does A completely eliminate the functionality of whatever the enzyme is doing, or it only hinders it slightly. All those things also play a role in all of these things. Let me before we move ahead, let me actually give you next week's assignment. So no class on Friday. I'm traveling, so we'll next meet on Wednesday, and we'll see as we go along if we do need to do an extra class to sort of make up for this last class. But for next Wednesday, I would like you guys to think about the following question. Let me just pull this up. Oops. So consider this as part of a fitness landscape. This is a huge fitness landscape, but we are only looking at a very small part of the landscape near the top of the fitness peak. Fitness peak is represented by a particular sequence whose fitness is 2. Our starting, the ancestral population that we are going to be starting with, think bacterial population, has a fitness equal to 1. Starting from this population of fitness 1, there are 2 evolutionary paths leading to the fitness peak. In the first path, you acquire first mutation, which gives you a fitness of 1.3, and then you acquire the second mutation, which takes you to the fitness peak. And that's the red one. Let's call it path 1. In the other path, the first mutation you acquire is one that confers a fitness of 1.6 to you, and then once you go up in fitness, you acquire the second mutation, which takes you to the fitness peak. Right? Remember that all 4 mutations are single nucleotide mutations. So none of this is, so each is equally likely. Each of these 4 steps, the probability of occurrence of each of these 4 steps in an individual is the same, because it only refers to one nucleotide change at one particular location in some context, right? So I think all of you will agree that if I were to start this experiment from here, I will end up here. No question about that, right? I start at fitness 1 and I'm going to end up at fitness 2. That everybody agrees? Yes, no?
Yes,
Yeah.
sir.
Yeah.
All right.
Yes, sir.
If that is the case, so my interest as a student of evolution is the following, that if I were to start this experiment from here, I want to know which of the 2 paths does evolution take to reach the peak. And when I ask a question like that, it is not sufficient for me to do the experiment only once. I will do this experiment 100 times, right? We can make it 1,000 or a million, whatever, right? It's statistically very large number of times. We start this experiment 100 times, and I simply want to, and I think we can all agree that if we start this 100 times, we will end up there 100 times, because natural selection is going to prevail. The question that I would like you guys to answer on Wednesday is that, is one of these 2 paths more likely than the others? And I don't mean, so that's why I'm saying if 100 is too small, we can make this a million or whatever. But are both these paths equally likely for the population to reach the fitness peak, or one of these paths is more likely than the other? Is the question clear?
Yes, sir.
Yes, sir.
Okay. All right. This is, I want you to think about this. Any thoughts?
I think the second path is more likely,
Okay, the
in
green one,
which
the
the
green
first
one.
mutation, yeah, green one.
Uh-huh. Why is that?
Because if we see at the relative fitness achieved after each mutation,
Hmm.
it's larger in the green path compared to the
Well,
red.
the first mutation has a larger benefit in the case that you are talking about, right?
Yeah.
So the effect of first mutation is larger here. The effect of second mutation is larger here. So what you're saying is that because of the fact that the first mutation is larger here, this is more likely.
Yeah.
But this is also a single nucleotide change, and this is also a single nucleotide change. So why would you say that?
Uh,
You
Agreed.
understand
It's
the
a
question?
yeah.
Yeah.
I understand.
Yeah.
Agreed. It is a single nucleotide change, so likelihood might be
Likelihood is the
same.
same, right? So
Yeah.
imagine that there is a A here, that A can change to T and you come here, that A can change to G and you come here,
You're right. But
right?
the distribution of the mutation in the population might be different, right?
What do you mean distribution of
I
mutation?
mean, if there are say 100 new individuals in each generation, out
Hmm.
of these 100, the green mutation might be seen in majority of the population.
Why do you
That way,
say that? Because no, what I'm saying is that there is at a certain position on the DNA, there is a A.
okay.
If that A changes to T, then you come here. If that A changes to G, you come here. Everything else on the sequence is irrelevant,
Okay.
right?
So
So
can we calculate the slope of each trajectory and see?
yeah, I mean, in one sense, this is, I'm sorry, in one sense, this is the slope, right? Because going from here to here is one mutation. So
Hmm.
on the sequence space, the distance is only one mutation unit, right?
Yeah.
And the y-axis change is 0.3 in this case and 0.6 in this case, right?
Yeah.
So remember that the Hamming distance between this sequence and this sequence is 1.
Hmm.
Hamming distance between this sequence and this sequence is also 1,
Yeah.
right? So in that sense, so slope is sort of 0.3 of this one and 0.6 of this, 0.6 in this case.
So the fitness value of green path, the first mutation being high, it might get fixed quickly than the other mutation might be lost. So from there it will reach the next mutation. Like
Why?
before next mutation occurs, this might get fixed.
Yeah, yeah, absolutely. That's that, and one other aspect plays out, but we'll discuss that. But you're absolutely right. Did everybody get that? So what Namrata is saying is that the chance of, and Namrata, you can correct me if I'm wrong. What the argument is that this mutation or this mutation has an equal chance of occurrence because both of them are single nucleotide changes. Now let's imagine that this mutation occurs. Then what you have is n minus 1 individual, and this mutation is only going to occur in one individual. What is going to happen at that instance is that you have n minus individuals at this fitness and one individual at this fitness. Now we are going to have time tau 2 that Anirudh was talking about, and this tau 2 is the time reached, the time needed by this particular mutation to reach fixation, so that everybody in the population has this particular sequence, and this is completely eliminated. That this process of the population shifting from here to here is tau 2, that time. In that time tau 2, so now what Namrata is saying is that tau 2 for this mutation going to fixation is greater than tau 2 for this mutation going to fixation. Does everybody see that?
Yes, sir.
Yeah.
Right? So while this is reaching fixation, there is a chance that this mutation might come up in another one of the individuals of this group, whoever is remaining here. If this particular individual, if this particular mutation comes up, then what is going to happen is that you have three different genotypes coexisting in the environment. You have original, you have these guys which are increasing in frequency, but you also have one new mutant, which this one which has come up. And should this mutant come up before this has gone to fixation, then this will never reach fixation, and this will be the evolutionary trajectory that the population will be taking. Does that make sense to everybody?
Yes, sir.
Right? As a result of that, and there is one more factor that we'll look at. As a result of this and one more factor, because remember we said tau2 is relatively small. Well, we'll look at the relative numbers. So tau1 is small, but we'll look at those how the interplay between those variables happens. But because of this reason and one more reason, the green path will be the more frequently taken path. So yeah, no need to submit this now. We've sort of discussed this. All right. Any other comments? I want to discuss a couple of other quick things. Any other points?
So the observation from paper 1 that the fixation time is longer than introducing a new mutation, that is always true?
No, again, I think it's going to depend on the population size, mutation rate, right? And the second mutation, how large is the region where the second mutation could occur? If you're talking of one, if you're occurring of one specific mutation, then it's going to take a long time. But if you are saying that of these 15 mutations, any one of them could happen, that of course changes the probabilities. But that's something that we'll look at in a couple of weeks, I think.
Okay.
All right. So let me ask you the following question. Imagine a large population of diploids. This is sort of switching gears, and I want to go back to diploids. Imagine a large population of diploids. At a particular locus, two alleles exist, capital A and small a. Frequency of capital A is X, frequency of capital Y, small a is Y. X plus Y is obviously going to be equal to 1 because at this particular locus, no other alleles exist. Selection does not act at this locus. So what is this is referred to as selectively neutral, this particular locus. It doesn't really matter as far as your fitness is concerned, whether you are carrying capital A or small a. Now, excuse me. I guess I forgot to put one more thing here, that capital A is dominant over small a. If that is the if that is the case, how does X plus Y change with generations? This should be X and Y. I wrote P and Q first, but then for later work I chose P and Q, so I then changed it to X and Y. This should be X and Y. How does X and Y change? What is the relative proportion of these three type of individuals? And is there an equilibrium that is reached in the population such that the relative numbers of these three type of genotypes in the population does not change as we go from one generation to another? Right. Does this question make sense?
No, sir. Can you rephrase it?
Yeah. So large population, at any locus, you have two alleles, capital A and small a. Frequency of capital A is X, frequency of small a is Y, and hence X plus Y has to be equal to 1. We are going to assume that this locus is selectively neutral. That means it doesn't matter which allele you are carrying, your fitness doesn't change. Now, the question is that what is the relative proportion of these three type of individuals in this population? Okay, is there an equilibrium such that maybe you will have given x and y, suppose this was 50-50, the frequency of the two alleles was 50-50. What is the relative proportion of AA individuals to capital A small a to small a at equilibrium? What we mean by equilibrium is that suppose after a long enough time, the relative proportion is 30%, 40%, 30%. If that is the relative ratio of these three genotypes and they are in equilibrium, what that means is that in next generation also this will be 30, 40, 30. It doesn't change with time. Does such a population take an equilibrium ratio? And the premise for this comes from when Mendelian genetics was discovered, and it was found that one of the alleles is often found to be dominant over the other allele. Then the question was that at equilibrium, will the allele which is recessive in nature exhibit itself at all, or will the entire population will be heterozygous and hence only the dominant allele is going to exhibit itself? Does that make some sense at all?
Yes, sir.
Yes, sir.
The sort of the question is that you have A and A at some frequency. Let me simplify the question and strip off the unnecessary details. You have two alleles at a locus, capital A, small a, at some frequency, no selection, right? And we want to know that this population is well mixed and anybody can mate with anybody. Under these circumstances, is there an equilibrium between these three type of genotypes? And by equilibrium, we mean that under all these conditions, no selection, capital A dominant over this, that, under all these conditions, is there an equilibrium between the three type of genotypes? And by equilibrium, we mean that going from one generation to another, the relative ratio of the three type of genotypes does not change. Okay. This problem, we are going to approach it like this. We need to make a certain assumptions, and let me finish this derivation, and then we'll talk about the relative importance of what this analysis is all about. The first assumption we are going to make is that there is random mating in the population. And by random mating, I mean that if I am a member of this population and I am choosing a mate, the choice of my mate is independent of what type of genotype is the individual at this particular locus. So we're not concerned about what is the genotype of the individual that I'm looking to mate with. So if this is the population structure, that this is me as an individual, and outside of me there are 9 individuals of AA genotypes, 7 individuals of capital A small a, and 3 individuals of small a, then the probability that I will mate with AA is just 9 by all the individuals in the population, that's 19. Similarly, 7 by 19, 3 by 19. This is called random mating, that the choice of my mate is not dictated by what is present at that particular locus or anywhere else for that matter. Right? That's random mating. The other assumption that we're going to make is that generations are non-overlapping. And what that means is that we have a population of size n, and that population is exactly replaced by another n of the successive generation. There is no overlap between generations. And of course, I mean, as we go through these assumptions, we will find that clearly they're all wrong, but it doesn't matter for reasons that we'll come back to once this analysis is done. Third mutation, no selection, no migration. What that means is that there is no force which is changing allele frequency. Remember, we said that we are going to look at evolution as just change in allele frequency. Anything that changes allele frequency is evolution. And the forces that change evolution, that change allele frequency are definitely selection, migration, because it can bring in new alleles, or if people are leaving, then it can take away existing alleles, or mutation, which leads to introduction of newer alleles. None of this is happening. So A and A are the only alleles existing and they're not changing. Then we are going to assume that population is extremely large. Okay? What that means is that statistically, if I'm looking at males in the population, I mean, this is males, females, let's say imagine a human population. If I'm looking at males in the population, let me assume that the frequency of capital A in males is P, and the frequency of capital A in females is also P. Population is large enough such that statistically speaking, when I sample the population into two groups of males and two groups called males and females, the relative frequency of the three types of genotypes in both the groups are identical because the population is extremely large. And capital A small a is 2q. Remember, these are the frequencies. So this is sort of the fraction of individuals which belong to each of these genotypes. Small a is R. Males, females separated, and hence P plus 2Q plus R is going to be equal to 1. This 2 exists only for mathematical convenience purposes. So it doesn't really matter if we take this as Q and not 2Q, but taking the 2 here just makes things a little bit easier as we move ahead. All right. If this is the existing structure, then the question is, what happens in the next generation? And to find out about the structure of next generation, we're going to look at it like this. First of all, we will find that how can an AA individual be produced in the population? AA individual can be produced if a AA male mates with a AA female, which can happen with a probability P times P, right? And from such a mating, any offspring that's produced is going to be an AA individual. So the percentage that the offspring produced from this particular mating will be capital A. That percentage is 100%. Hence, the total number of AA individuals produced from this mating is simply going to be equal to P times P, which is P squared. But an AA individual can also be produced if a capital A male mates with a capital A small a female. The probability of that happening is obviously P into 2Q. But from these matings, only 50% of the individuals are going to be capital A. The other 50% are going to be capital A small a, and we're not concerned about those individuals here. So that's only going to be, only half of these individuals are going to be capital A. So the total frequency of capital A offsprings produced from this second row of matings is going to be P times 2Q times half. So this half and 2 cancel. That was one of the reasons why we put 2 here typically in this derivation. And you get PQ. Similarly, the other way around, that a male can be capital A small a, female can be capital A, and you get the same answer. And the last way could be that you could have a heterozygous male mate with a heterozygous female, and obviously only 1/4 of those matings are going to result in a capital A individual, and that's just simply Q squared. So this 1/4 cancels the 4 here. What that tells me is that frequency of an AA individual is going to be, I can simply add these up, and this is the frequency of an AA individual. When I'm doing a similar exercise for small a, it's just a mirror image of the same thing. If I replace P by Q in this entire analysis, I get the same thing. And this is the frequency of the small a individual in the next generation. The bookkeeping is a little more longer when I'm talking about the small a, capital A individual, right? So small a, capital A can be can be born out of a mating of capital A, and capital A, small a, 50%, and so on and so forth. I mean, you get the picture. It's pretty straightforward, right? So this mating obviously is, I wrote this by mistake, but small a mating with small a is obviously never going to give you a capital A small a, because mutation is not allowed. So basically you strike this off. So there are 7 different ways by which you can produce a capital A small a individual. And we sum these up and we get the frequency of capital A small a individuals. What this analysis tells me is that in the next generation, the relative frequencies of the 3 genotypes are going to be this. And again, if we can add these up, and that comes out to p plus 2q plus r, which is a whole square. But as we defined here, the way this was defined, that p plus 2q plus r was actually equal to 1. So in the next generation, you obviously have frequencies which sum up to 1. Right? So that's just 1 square. So I can, if given the frequencies in one generation, I can calculate the frequencies in the other generation, in the next generation. Now, what I'm interested in is that what happens to the allele frequency. So remember, this was not the allele frequency. This is the frequency of the different genotypes that are existing in the population. To get the allele frequency, suppose I'm interested in the allele frequency of capital A, then the allele frequency of capital A is going to be how many A alleles are present in the population. All of these individuals are carrying 2 copies of capital A. So I multiply their frequency by 2. So I get 2 times p plus q square, right? Plus all of these individuals are carrying 1 copy of capital A. So I multiply this frequency with 1, right? This gives me the total number of capital A alleles which are present in the population. I divide this by the sum of all these, the sum of all the alleles which are present, which is this times 2 plus this times 2 plus this times 2. That's the total number of alleles that are being carried. Not total number of copies of all the alleles that are being carried in the population. So this times 2 plus this times 2 plus this times 2, which is basically just 2 into 1 square. So that is just 2. And this simplifies to p plus q, p plus q times p plus 2q plus r. But I know that p plus 2q plus r is simply equal to 1. Hence, frequency of allele A is actually equal to p plus q. Right? Does this make sense at all? Is what we're doing make sense at all?
Yes, sir.
Others, questions? Yes, no?
It's fine.
Okay. Anirudh, go ahead.
No, I say it was fine.
Okay. The point being that originally we started, this was the frequency of the 3 genotypes, and now if we originally we started with this, right? And if I were to ask you that what is the allele frequency of capital A here, the allele frequency of capital A is, remember, this is the original population, not the subsequent generation. This is the frequency of allele A in the subsequent generation that we've calculated. In the original population, we have the allele frequency of A is 2 times P, because these P individuals are carrying 2 copies of capital A. So in the numerator, we have 2 times P plus these 2q individuals are carrying one copy of capital A. So in the numerator, you get 2p plus 2q, and in the denominator, you get 2 times p plus 2q plus 2r. But p plus 2q plus 2r is simply equal to 1. So the whole expression, the allele frequency of capital A, comes to 2p plus 2q divided by 2, which is just p plus q. And as we see here, that in the next generation, the allele frequency of capital A remains at p plus q. Again, I mean, I probably should have done this better, but I understand if this doesn't make complete sense. But the point to note here is that what this shows is that allele frequency going from, in the scenario that we have described here, allele frequency does not change. And it shouldn't change because there is no selection acting, and there is no selection, there is no migration, there is no mutation. Hence, allele frequency does not change from one generation to another. What does happen, however, is that given a certain allele frequency, the population. Let me do this with an example. So let's imagine this. Let's imagine this, that originally you had a population AA, which was 50%, and AA, which was 50%. Right? Let me ask you guys, what is the frequency of allele A? Anybody? This is my original population.
Half.
Yeah, so we can simplify this even further. We can say 50 individuals are AA, 50 individuals are AA. There is no heterozygous individual. What is the frequency of capital A? Half, right? So frequency of allele A is half. Frequency of small a is also half. Right? Now the question that we are asking is that when this moves to next generation, right? What is going to change? Is this going to change, or is this going to change? Is anything going to change at all? And what is your answer?
The genotype frequency will change. The allele frequency won't.
Exactly, right? So as you go from one generation to another, the allele frequency will not change because there is no selection acting. However, it's clear to see here that if mating is random, right? If I choose to mate with someone independent of what their genotype is, clearly there are going to be heterozygous individuals which are produced in this population, right? So what is going to happen in the next generation is that this will become 25, this will become 25, and this will become 50. Right? And frequency of the alleles is the same here. Frequency of alleles doesn't change because there is no selection acting, but these alleles, these 50% alleles existed in one particular structure of the population on the left. However, when we allowed for random mating, then these 50% alleles rearranged themselves in the population such that these 50% alleles were distributed in a different way across the three genotypes that existed in the population. And if we go from here, if this is our starting population, and if we go to the subsequent populations here, what we will find is that this structure of the population, this red structure of the population, doesn't change anymore. So what has happened is that this 50% alleles existed in the population in a given fashion, they were not in equilibrium. They were not in equilibrium because I can clearly see that if a capital A mates with a, small a, which is going to happen, you're going to produce a heterozygous individual. So there are going to be heterozygous individuals which are not there in the population at this point in time. And if I were to allow everybody to mate randomly, what is going to happen is allele frequency won't change because there is selection, but the three genotypes will quickly attain a frequency which is at their equilibrium. That is, going from one generation to another, that allele frequency will not change. This equilibrium is called Hardy-Weinberg equilibrium. Does that make sense?
Yes, sir.
Hardy-Weinberg equilibrium, because of the assumptions that we made that went into it, doesn't make any sense whatsoever. You cannot have extremely large populations. You cannot have matings which are independent of genotype. You cannot have a scenario where there is no migration, no mutation, no selection. All of these forces are going to be operating. However, Hardy-Weinberg equilibrium is still majorly used in all population genetics and evolution model, because it tells me that if evolution wasn't happening, right, if all evolutionary, remember again, evolution is anything that brings about changes in allele frequency. If everything that was bringing about changes in allele frequency was removed, my population should be here. This should be the distribution of the population if everything that brought about changes in allele frequency was removed. Now this becomes my baseline. Now I'm going to study whatever context it is, that whatever population in whatever context I'm looking at, and I'm going to study its distribution, and I'm going to comment on how far or how close am I to this Hardy-Weinberg equilibrium. So in itself, Hardy-Weinberg equilibrium is perhaps not very useful because it's an extreme exaggeration of a scenario, but it gives me a very important baseline to compare things with. The other thing is that Hardy-Weinberg equilibrium was proposed by two different people, Hardy and Weinberg, in 1908, and it was done with a different context. Post rediscovery of Mendel, people thought that the dominant allele will spread in the population, no selection acting, but there was a debate among biologists that the dominant allele will spread in the population to extent that an overwhelming majority of the population will be carrying at least one copy of the dominant allele. Hence, the physical manifestation of the individual is going to correspond to the one that matches with that of the dominant allele. And the recessive phenotype is going to be not expressed in the population at all. Hardy-Weinberg says that that's not true. There is going to be a fixed number of individuals, small a, which are going to exhibit the recessive phenotype, and the number of those individuals is going to depend on the initial allele frequency in the population. If selection is not acting on this allele, then you may have a fair number of individuals which are actually exhibiting the recessive phenotype. I mean, it sounds trivial, but it wasn't at the time. Hardy is the mathematician who was Ramanujan's mentor, and he was approached by Punnett. Those of you who are from genetics will know Punnett squares. He was approached by Punnett to solve this problem, that is there an equilibrium frequency of the three genotypes that can exist in the population? And at least the Hardy part of Hardy-Weinberg was coming up with the derivation which came to these equilibrium frequencies and clearly show that at equilibrium, the three genotypes are going to coexist together, and the frequency of these three genotypes is not going to change one generation to another, assuming all those assumptions are somewhat representative of a real ecological niche, which may or may not be true, but that's not the point. The point was that they're going to coexist. Today, Hardy-Weinberg equilibrium is used in the context that we discussed. That Hardy-Weinberg equilibrium gives me a baseline, that this is what the structure should look like if everything that brought about evolution was absent from the context in which I'm looking at this. and then I see that what is the actual distribution that I see in the field or the experiment, and then I draw my conclusions from there. All right. Let's stop here. We have just a little bit to do. Is that now we want to introduce selection into the media. Mutation, migration, and selection were the three forces that we ignored in Hardy-Weinberg. We want to start by introducing selection into it, and then we will introduce mutation into it. Migration is more ad hoc. It's difficult to implement in any structured, logical way. But we're running out of time. We can take a few questions. Otherwise, I will just see you guys on Wednesday now. Yeah, questions, comments, anything?
Sir?
Yeah. Yeah, yes, go ahead.
So my question actually is, a dominance of an allele is dictated by the selection acting on the population, right? So how are they coming up with the dominant and recessive allele without considering selection at all?
No, so let me correct you there. A dominance of an allele is not coming out of selection. Dominance and selection actually have nothing to do with each other. Dominance is only coming out. Suppose, again, I'm making up this example. Suppose I'm carrying one allele which produces a pigment which gives blue color to my eyes. Okay. But I'm carrying the other allele which gives black color to my eyes. Right?
Okay.
Now, when the pigment is produced in my eye, the blue is completely overshadowed by the black.
Right.
Right? So, hence, as far as physical manifestation is concerned, I am only going to exhibit blue eyes if both the alleles that I'm carrying are blue colored pigment alleles. Right? Right. So the black allele is dominant over the blue allele, but that has nothing to do with selection, right? I mean, it doesn't change my fitness in any way. I mean, if it changes mating behavior, it changes, but in a very general sense of the word, it doesn't change the fitness of the individual. So dominance is more related to biochemistry, and we will discuss this the last class of. We are discussing modern synthesis, and the last class of modern synthesis, or maybe two classes of modern synthesis, will be debates that occurred between Wright and Fisher. And we will discuss two, they differed on many different points, but we will discuss it from two aspects. One is a very general difference of opinion between the two about how evolution happens and how populations behave on a fitness landscape. And the second debate is going to be the mechanistic basis of why there should be dominance. Okay. So dominance has more to do with biochemistry rather than selection. It could impact selection, but it doesn't dominate. Just because dominance exists doesn't mean that it will there will be selection.
Got it.
Yeah. Other questions? Okay, great. I will see you guys on Wednesday. Have a